flat. You can see this is actually normalized at 440. So here's a very strong peaked absorption. Here's some flatter absorption. So you can begin to look at the shapes of absorption based upon the packaging of that pigment. And actually, I'll talk about this later. Okay. So phytoplankton have a lot of variability, and that's what, so Mary Jane and Ivona are going to spend a lot of time talking about the details of that, so you'll hear that all again. Other organisms also have absorption spectra. Some of them have this nice exponential that's sort of reminiscent of CDOM, probably because it's very similar compounds in the dissolved and particulate fraction, but they may have cytochromes in their, the cytochromes. Oh my God, in the mitochondria. And so those have absorption peaks. You can identify those. Those aren't pigments. This is some heterotrophic bacteria that also have some of these peaks, but again, this background absorption by organic matter. This was that same microphotometry for detrital particles. Again, individual detrital particles. He measured every detrital particle in the sample. You can make detritus. Here's an example of taking phytoplankton and just exposing them to natural light until you basically are degrading them with light and you're starting to turn them into a detrital particle. If you look at the slope of that exponential of non-algal particle, the slope is much flatter, 0.011 compared to 0.016 for CDOM. And again, that same distribution of the most likely slopes of the exponential found in the European waters, again, is about 0.011. So different from CDOM, which allows you to separate absorption by the non-algal particles from CDOM, because CDOM is a much steeper slope. Here's an example of looking at the absorption spectrum of particulate matter that doesn't have phytoplankton in it, marine and freshwater samples. Again, this nice absorption, exponential absorption. But if you notice there's features in here, it's not quite as smooth as the CDOM that we see. There's shoulders. And this is from a PhD work from Mega Stoppa, who actually got her PhD here with Larry Mayer. And her thinking was that this has to tell you something, these little tiny shoulders. And so what she did is she did a derivative, I think she did a second derivative, of each of these spectra. And this is the associated second derivative. And so it's identifying the changes in slope from that exponential. And what you see are these very distinct peaks. And it turns out that those are associated with absorption peaks of iron oxide minerals. And so you could actually begin to say something about the iron content of that particulate matter on those sediments. So again, the absorption, there's a lot of variability, natural variability in the absorption in this particulate component, but you can pull out other specific characteristics about it from its absorption. So anything that causes variability in your sample is an opportunity to extract more information out of that sample. Okay. Other inorganic particles. These are two different studies, one by Baban and Stramsky, one by Patterson, where they looked at the absorption of different minerals. And you can see, again, there's this sort of overlying exponential decay, but again, there's lots of features on here, again, probably due to some iron oxides. This is on a log scale. This would be the exponential approximation of the exponential slope. And so looking at the variations in that slope would tell you something about the composition of the mineral samples that they were looking at. So those are definitely showing up in your non-algal particle measurement of absorption. So we have inorganic particles, we have organic material, we have pigments in phytoplankton, we have CDOM, we have inorganic salts. So when you add all of those up, you get total absorption. So here's just an example of water plus non-algal particles, phytoplankton, CDOM, and that purple is your total absorption. And this is from a study, this is from Morel and Pure, 1977. It's one of my favorite optical papers. We'll be talking about it more later. But these are examples of the total absorption spectra for different waters. And they came to describe these as sort of regions which are dominated by water, regions where the absorption has this U-shaped characteristic, upside down, or this U in terms of the spectrum. These were dominated by inorganic particles, which have that exponential absorption. It probably also had some organic absorption in here. And then these V-type absorption spectra, which were found when phytoplankton were a large component. And so they tended to describe the shape of absorption based upon which component was dominating. And so by looking at the shape of the total absorption, you can begin to deconstruct it with your eye, what is dominant. If you have a nice exponential in the blue, it's probably CDOM, or non-algal particle absorption. If you have a lot of features in your absorption, you're probably dominated by phytoplankton. And so as you begin to collect absorption measurements, you will start to see patterns in the shape and the spectrum of those scans, and that will give you an intuition about what's in the water. And so if you're working with AC meters and you're profiling with them, often we put the graph up that shows us what the trace with depth looks like so we can see where things are distributed in the water column. But you may want to be looking at the spectrum display so that you can get a sense of what that spectrum looks like as well. And we'll do both in the lab today. Okay. Any questions about constituents, absorption spectra? Nope. Okay, there will be a test. No, just kidding. Okay, so you're going to hear a lot more about phytoplankton and their absorption properties today. We will be making measurements of CDOM absorption in the lab, both in a spectrophotometer and with AC meters. And then we'll talk about particular absorption methods in the lab tomorrow. So this isn't all you've heard on absorption. So let's see, 9:30. I'm going till 10, Emmanuel? Okay. Okay, good. All right. Did I talk too fast? Okay, there's a question, yeah.
Quick question. Those like absorption peaks for when you're looking at different pigments and stuff, are those also indicative of pigment concentration as well?
Yes. So if you think about I like to think of measurements in terms of orders of variability. So if you're measuring absorption and you see an increase in absorption, that is primarily driven by an increase in concentration. So order 1, you got more stuff in the water, you got more absorption. So when you see big relationships between, say, the absorption concentration versus chlorophyll, or the scattering concentration versus chlorophyll, and you get this line with a lot of scatter on it on a log-log plot, that's your order 1 variability. The concentration of material primarily determines the magnitude of any of your optical properties. To second order, when you start to see variations in the shape of the curve, that's telling you information about the composition of that. So if you look at the absorption spectrum of phytoplankton and you see different peaks and shoulders, that is primarily driven by the presence of different pigments. If you look at the relative amounts of, say, a pigment that's absorbing at 560 nanometers versus chlorophyll absorbing at 440, and you see variations in that ratio of absorption, that's telling you you have variations in the relative concentrations of those two pigments. So you can begin to look at, am I looking at a variation in magnitude? Am I looking at a variation in shape? Am I looking at smaller variations in relative shape? So we use that information to tell us a lot about what's in the water. And so the more spectrally resolved your absorption, the more you can begin to resolve more than just concentration, more than just pigment composition, potentially taxonomy, potentially packaging, light history. Turns out they also change their nutrient ratios if they're nutrient stressed. So there's a tremendous amount of variability in the absorption spectra, but every time you observe variability in an optical property, instead of going, oh, go, oh, that means I can resolve something more. I can pull more information out of that optical measurement. Okay, so we will be, in the lab, we will be working with absorption meters, AC meters, and hopefully we'll get an ICAM set up in the next week or so. We'll also be doing discrete samples in the lab using quantitative filter technique for the particles. Today we'll be doing CDOM. But I wanted to talk a little bit about the quantitative filter technique because you'll be doing it today, and just so, or not today, tomorrow, just so that when you get into the lab, I mean, you're going to be doing a lot of stuff in the lab, so I want you to be thinking about it before you get into the lab. So again, imagine you take your water sample, you filter it. Today you're doing CDOM, tomorrow you're doing absorption by the particles. So filtration is that first order of processing that separates the particles from the dissolved. The other thing that it does is it concentrates the particles from a relatively dilute medium onto a concentrated sample. So if you're in waters that have very low concentration, you can filter a lot of water to get a good signal, which is sometimes very difficult to do in situ because you only have a certain path length for measuring absorption. So with any measurement that you would make spectrophotometrically, you need to have a reference. So we'll be talking about this with regards to AC9. What is our reference material? And in a spectrophotometer, Mary Jane, are you doing dual beam or single beam? Single beam. Okay, so first you would measure a scan of your reference material. Then you would measure a scan of your sample. So if you have a filter with phytoplankton on it, or any sample filtered onto it, what part of that are you not interested in? The filter. So you want to remove that signal. So you take a measurement of a filter, right? So you'll want to, sometimes you can just take a bunch of readings of blank filters and figure out what the average filter is. Some people do that approach. Some people measure a single filter and use that as your blank. A baseline is where you actually are measuring some of the signal associated with the fact that if you had nothing in your spectrophotometer, you still get a signal. And so it has to do with the variation in light. Intensity has to do with the gratings that give you wavelength. And so you'll be taking a baseline scan as well. But then you have this reference scan with your blank in it. So thinking about what your blank is going to be for all of your measurements is really important. So this is the conversion from what we call optical density or absorbance. If we've set our spectrophotometer up to capture scattered light, it's measured in log base 10 units, and we're converting it to natural log units. So you have to put a 2.303 in there to do a conversion between the two. And it's the loss of absorption. So this is that phi incident minus phi transmitted from yesterday, divided by the path length, will get you absorption. And so what is the path length of your sample if you've put it on a filter? So when I showed you yesterday, I had this little infinitesimally thick layer of water, and that was our path length. But now I've taken that water and I've put it on a filter. So, I mean, you could go in here on the filter and you could measure the micro layer of particles on there, but is that what they were when they were in the water? Well, let's think about taking a volume of water, and if we were measuring the absorption, let's say the light through there and then the light transmitted, this would be our path length, right? But now what we've got is this filter, and we have filtered a volume of water through it. And all of our particles are now going to be on that filter. So you can take this volume of water and you can shape it in your mind to look like this, where the area of this cylinder on its end is the effective area of the filter, right? And the height of that volume of water would be the height of taking this volume of water and reshaping it into a volume of this area and this height. So what's your path length? If we hadn't filtered yet, here are all of our particles. That is our path length. Pardon?
The volume, the total volume divided by area. But at the same time, we can conceive the different cylinders.
So you can imagine different cylinders, right? But what you're doing is creating a cylinder. So yeah, because your filter is only this big, right? And you've got a volume of water. But what you're essentially doing is taking that volume of water and squishing it into a really, really tall column that has an area the size of your filter, right? So in reality, this looks more like this. Right? This volume equals this volume equals this volume, if I were cleverly drawing.
Or it would be not the area of the filter, diameter of the filter, but diameter of the part with the particles.
Exactly. The effective area. That's your path length. Okay? So, that's exactly what I've done here. So, the volume filtered is the effective area times the height, and height is your path length. So that's what's going to go into your equation here. Okay? So that's in your handout. Now, of course, we want absorption per meter, so you have to convert this to meter. And so, if you're measuring in mils, that's cubic centimeters. And if you're measuring your area in centimeters, you've got centimeters, and you've got to convert it to meters. And so that shows up in the equation. You'll see that in the handout. Okay. So I just told you that by filtering onto this filter, it's really nice because it increases the concentration. I mean, you can go from a path length like this to a path length like this, which is quite helpful. But the filter, how many people have seen a glass fiber filter? It's what we use for chlorophyll analysis. Most people have, right? So what do you notice about the glass fiber filter? What do you think its optical properties are? What color is it? White. Do you think it's primarily absorptive or scattering? Scattering. So if you imagine you've got this filter with all of these different particles and you shine a beam of light on it, if you've ever looked at what happens when you shine a beam of light on it, you get scattering in all directions. But you're also getting scattering within the filter, which is increasing the opportunity for absorption by the particles, because the light doesn't just go through once and come out. If it's scattering, it's increasing its optical path length. So you have to account for that. And so, Okay, so it creates a pretty uniform light field, which turns out to be kind of interesting and clever. Well, it can be. You can make use of that. But the most important thing is that it increases the optical path length, and you need to account for that. And so there's been a long history of figuring out how to account for how much increased path length through your sample happens on a filter pad versus having it be in solution. And so some of the latest approaches have involved something called an internally mounted integrating sphere. And this is the basis of the ICAM, but in the spectrophotometer, you can have a light incident on your sample, and this is a sphere, which is highly scattering. Here's your detector. It's measuring the amount of light coming through some portion of that sphere. You can figure out what it would be if you could detect all of it. And so if you took a sample and you were able to measure the absorption of light going through that sample, where you're essentially collecting all of the scattered light, it becomes a more optimal absorption meter than if you took a sample and put it in what we call transmission mode, which is here's the incident intensity, here's the transmitted intensity, but you know you're losing scattering, which of course you're going to call absorption, but it isn't. So by internally mounting, you can do a much better job. You can actually put these filters in an integrating sphere, and that seems to be where the field is moving, because then you can collect all the scattered light from the filter. But let's say that you could measure the absorption of your suspension in the absence of scattering. That would be truth, close to truth. Then you could take your sample and filter it onto a filter and measure it in your spectrophotometer, and it's going to be different. The absorption that you determine is going to be higher because you have all this multiple scattering by the filter. But if you knew the sample was the same, you knew the answer should be the same. So you could look at the ratio between truth and your filter pad. And the ratio would tell you the difference in the optical path length of the photons caused by scattering by the filter. That is essentially the approach of trying to figure out what path length amplification is. Amplification of the path length due to scattering by the filter going through these particles. And so this is determining this, what they call the beta effect or the path length amplification effect, by looking at the ratio of the absorbance on the filter or optical density versus that in a cuvette, gives you the ratio of the optical path length versus the geometric path length. Geometric path length is this. Optical path length is the increased path length caused by multiple scattering in your sample going through each of your particles more than once. So this is from a paper that is in press from Dario Stramski's lab, where he looked at the absorption measured on suspensions of different minerals, cultures, detritus, natural waters, measured in the near absence of scattering, versus the absorption or optical density absorbance measured on a filter pad in transmission mode, which is what most spectrophotometers are set up to do. And historically, all of the measurements of absorption were measured this way. And you can see that this is the relationship here. And this is a linear scale. And so he has come up with a relationship of how to convert a filter pad measurement using transmission mode, which you'll be doing in the lab tomorrow, how to remove the effect of multiple scattering to retrieve the sample optical density. So you'll be doing these calculations in the lab. You'll also, because there's been a long history of beta corrections, and every lab that measured absorption developed their own beta. Because of the different ways that they did these measurements, they're all different. There's no consensus, which is a problem because all the data that goes into CBAS or other databases all have different beta corrections. So there's a lot of uncertainty in the absorption spectra that have been derived using this method. So we're going to have you use a couple of different beta corrections along with this one and get a sense of how different the absorption might be. So if you submit your absorption to CBAS, maybe you were on the same cruise, you collected the water separately, did your own analysis, but you did two different correction factors. That all shows up in these data sets. So what is that uncertainty? And as we look back through the historical archive of filter pad absorption, that legacy of different beta corrections is in there. So the community is really trying to move to consensus, and they recognize that the integrating sphere is preferable because of this, the lack of scattering correction, or not the lack of scattering, but the reduced effect of scattering in the integrating sphere, but these are very expensive and nobody has one. So can we use this to go back and recorrect archives of filter pad absorbance? Well, as long as we had the original absorbance, yes, we could. But whoever wants to do that job should raise their hand to go back and redo all of CBAS, right? So in the absence of doing that, which some people actually are going to do, but in the meantime, it's really important to understand the uncertainty in your measured absorption based upon different betas. And so that's one of the exercises you'll be doing today, is seeing, is it a factor of 2? Yeah, it is.
So what's the advantage of keeping the chlorophyll on a pad rather than dissolving the chlorophyll out into solution and then making
Ha-ha! What happens to the molecule when you take it away from, tracking me over here, take it away from the binding that protein with pigments into a membrane versus putting it into, it changes the molecular structure, it changes the peak at which it absorbs, it changes the harmonics, the relationship between the electronic absorption peak and the harmonic peaks. It changes the packaging, it changes the relative absorption between the pigments. So years ago, Bob Bidigary was using an approach where he looked at the concentration-specific absorption for each pigment. And then he would go out and collect water samples, and using HPLC would get the concentration of every pigment. He would multiply it by its specific absorption coefficient and reconstruct absorption spectra. And he would get beautiful curves that look like what you would measure if you measured an extract in the spectrophotometer. But it doesn't look like what the absorption is in a cell. And so there have been a number of people who have been like, oh, okay, well, we have to apply a shift. Okay, we have to apply a packaging. And so it ends up being a lot of modeling based upon what you know you should get when you have absorption in the cell. So it sets up another set of uncertainties. Yeah. Good question. Yeah.
So one of the comments about going back historically and trying to correct things is made more difficult by the fact that people were not rigorous in how they documented their protocols. So one of the things in order to do this is you need to know what the geometry is of the spectrophotometer. For example, historically, I used to have an Amenko-Bowman spectrophotometer that had a beam scrambler. And so some of that scattered light was collected by this long rectangular path that had a mirror. And so the light exiting the slit would get bounced around, and it would be then, it would hit the photomultiplier tube. And so you would capture some of that scattered light.
It captured all of this.
So the beta correction for that versus the carry 50 that you'll use tomorrow is going to be really different.
Yep.
So you need to have the type of spectrophotometer, the model, exactly how you place the filter. And so this whole idea of traceability, I think as a community, have become a little bit more mature in terms of the importance of tracing what you may think is a trivial step, but actually becomes a really important component.
Absolutely. And that's exactly why there was no consensus with this path length amplification, because people were trying to compare this to the suspension, but they didn't have an integrating sphere. So they put the cuvette here, and those particles were scattering light. But they said that that was truth. And I even read one paper which said that beta was 1. For beta to be 1, that meant that the path length through the filter was the same as the path length through the cuvette. So the cuvette had the same amount of scattering as the filter pad, which meant that they must have had a huge concentration of cells in their suspension to have this huge amount of scattering to match the particles on a filter. So what you know to be true was that they didn't have truth. They had an absorption measurement that had a ton of scattering in it, and they called it truth. Then they had a filter pad absorption, which they tried to account for that path length amplification, but they had just as much path length amplification in their cuvette measurement. And so you can find betas in the literature that range from 1 to 6 without any accounting of how they took care of the scattering either by the filter or by the suspension to which they were using as their truth. The other thing is that when people submit data, they were submitting absorption. And there's no record of the volume filtered, so you can't get back your original absorbance or optical density. So now what NASA is requiring when you submit data, that you submit the original optical density scan, the volume that you filtered, the beta correction factor that you used, and the calculated absorption values. That way, it will be much easier if consensus is reached about corrections. You can actually go and do just a giant reprocessing of that data set automatically. So more information is better, yeah.
So I'm a little confused. I haven't done the integrating sphere method. Is it only the cuvette, or do they look at filter?
So what Stramsky do, they derive a beta, a path length amplification, by doing a suspension in the integrating sphere, and they run all of their filters in the integrating sphere as well, because then you're capturing all of this.
Do they rotate it?
Yes. Well, yeah, so Darius. So they have a beta correction based upon measurements of the filter pad in the integrating sphere, but recognizing that the field also uses integrating spheres with samples mounted on the outside of the integrating sphere, which would be what we call transmission reflectance mode. They calculated a beta for that. And then they also calculated a beta for filter pads measured in transmission mode. And then they compared the beta that they derived for a filter in here versus a filter in here, or a suspension in here, and then compared it with all of the published beta corrections. And what you find is that there's easily a factor of 2 variability between all of the beta models that have been published.
So did you compare the AC9 with the integrating sphere?
No. That's what we're doing here.
Because that seems like,
Well, what it does is it provides you also with a sense of the scattering in the AC9.
Right. And so what I don't remember from previous classes, or whether you published this, is a beta correction based on an ACS measurement, because that's actually
I did. That was, yeah, I did in 98, yeah.
As part of the,
Yeah. Yeah, I did that. That was the 98 paper, yeah.
Yeah, and now there's a new scattering correction for AC meters
Mm-hmm.
From Rogers, he has an integrating sphere that he can flow through watering and through samples. And he has compared those measurements with AC meters. Correction that, particularly for samples where there's a lot of inorganic particles,
Mhm.
is much better
Yeah,
than standard bypass.
yeah. So,
All work in progress.
yep. And we'll be looking at those in the lab, because we'll have an integrating sphere that you can actually fill with water. This one is a laboratory sample that you can mount a sample in the middle, but the integrating sphere, the in situ one, the ICAM, you actually fill the cavity with water. And so then you don't have, you don't have particle filters, you don't have cuvettes, et cetera. But so this will be the exercise that you do tomorrow, and it's a lot of correction that you have to do because of this filter pad. And you're like, well, why the heck are we doing this? But it's because the filter pad provides you with the opportunity of separating the phytoplankton from the other particulate matter. And for people interested in phytoplankton, who are interested in being able to look at pigments, look at photosynthetic potential, et cetera, that decomposition by separating those components is really critical. And right now it is the only game in town. It's easy to separate particles from dissolved. You can just put a filter on there. You can put a filter on your AC9 or your ACS. But you can't then also put a phytoplankton filter. So, okay, I'm going to go ahead. One more question.
When you do the filtration, still there are some dissolved material on the filter. How do you
It's very small compared to the particles that are on the filter. Although if you're in a really, really high CDOM area, you can get some absorption on there. So sometimes people give it a rinse with filtered seawater that doesn't have high CDOM in it, and you can get that CDOM out of there. Yeah, so if that's your area, you have to rinse that CDOM off. Yeah, and I would also note that when you're filtering your sample, there's an optimal thickness that you want on your filter pad, and it just takes some practice. You need to have your optical density of, say, let's see, I need an absorption spectrum on here. Let's see. Okay. So you need, this is absorption, but if this was the optical density that's coming out of the spectrophotometer, you really want the majority of the spectrum to be between 0.1 and 0.4 optical density units or absorbance units. And obviously it's going to be lower here because there's just not much absorption here. But you'd really like to keep the major peaks within that range. If you have more material on your filter pad, it starts to get kind of muddy. And then you start having a lot of interactions between the particles on there and inefficiencies with transmission. If you actually convert your optical density to transmission, it's something like 0.1% of the light is actually making it through the filter pad. So you don't want to make that be 0.001% light making it through, because then you have a signal to noise problem. On the other end, you don't want it much lower than that because the optical density of the filter pad itself is huge. It's like 2. And so if you imagine you have variability from filter pad to filter pad on the order of 2, and your signal is on the order of 0.02, you've got a really very bad signal to noise ratio there. So optimally, try to figure out how much you need to filter to get within that range. Okay, and if you're going into the UV, and the UV is taking off and getting really, really high, well, you might have to filter a different filter so that in the UV range, it's between 0.1 and 0.4. So it's just something to keep in mind. And as you're going to do, I think you're going to filter different volumes, you'll see the difference in the color on the filter, and you'll also see differences in the noise, depending on if you're on the low end versus the high end. So, okay, I'm going to quit there and take a break until 10:30. Is that right?
Yeah.
Okay.
Or 10, maybe 10:25. So
10:25.
there's coffee in the food
Up in the hall, yeah. Right here.
I know.